Hello everyone, welcome to our YouTube channel Logic Medico. We are understanding the concepts in medicine. If you are new to our channel, kindly consider subscribing to our channel and don't forget to press the bell button for the latest notification of the video. Today's interesting topic is MCQs on coronavirus. Let's start one by one. Coronavirus belongs to which order of viruses? Your options are Bunyavirales, nidovirales, picornavirales, herpes virales, ortivirales. Your time starts now. If you are told option B, then you are told the correct answer. Coronavirus belongs to nidovirales order. Okay. Their order is, coronavirus order is nidovirales. Family is Coronaviridae. Please note the name Viridae. Subfamily is Coronavirinae. So there can be a question based on the family or subfamily. So you should know family ends by DAE and subfamily ends by NAE. If you just think subfamily, if you reverse this U, it will become like this N. So you can remember it like this so that it becomes easy for you. So coronavirus you will use N95. So nidoviral, that you can remember. Hope it helps. The second MCQ. What type of virus is coronavirus? Options are non-enveloped and enveloped, or both, which you can think. The time has started. You can see the virus right here. So it has got one covering, right? So it is an enveloped virus. So coronavirus belongs to nidovirale family order, then family Coronaviridae, subfamily Coronavirinae, NAE. It is an enveloped virus. Okay. This virus has got a lipid bilayer similar to the cell membrane of the human cell. Okay. So that is about the second MCQ. Coming to the third MCQ. Coronavirus has neutral sense, negative sense, positive sense, all of the above. We are talking of genetic material. Coronavirus has got what sense of genetic material? Your time is already running out. Is it neutral, negative, positive, or all of the above? The answer is positive sense. Option C is the correct answer. So what is this positive sense mean? Meaning coronavirus has got a genetic material which is mRNA, and this genetic material is a positive sense, meaning as soon as it enters the body, it directly produces or translates the proteins by the host ribosome. It doesn't have to do anything else. It just has to enter the body, enter the human cell, and directly starts producing its proteins, the various components of protein which we'll see later. So those proteins are directly produced. That is called positive sense mRNA. If at all it's a negative sense, it's not coronavirus, but I'm telling for completion. First, the virus has to replicate and then it has to produce its protein. But here it's not so the case. Positive sense, already it has got all the material, the mRNA. It can directly come and utilize the hosts, that is human ribosomes, and starts producing the protein what the virus requires. Okay. What type of virus is coronavirus? Single stranded RNA virus, double stranded RNA virus, double stranded DNA virus, single stranded DNA virus. Your time starts now. If you're told option A, then you're told the correct answer. Coronavirus is a positive sense single stranded RNA virus. So this positive symbol means, I just now told in the previous MCQ, positive sense where the RNA enters, directly it can produce its protein. It is single stranded. The RNA is not double stranded helix like a DNA. It will be single stranded, and it is a messenger RNA. So it can directly translate the proteins by the host by utilizing the host ribosomes. So coronavirus, here only you can see the answer, RNA. It is a single stranded answer. It's a single stranded. It's a positive strength. Okay, positive sense. Next, the genomic size of coronavirus is option 26 to 32 kilobytes, 32 to 38, 38 to 40. Sorry for that. It's 40, 44 to 50. This is 44. Okay, 38 to 44, 44 to 50. The time starts now. On an average, the genomic size of coronavirus is 30 kilobytes. So the one which is fitting to the 30 is this option, 26 to 32 kilobytes. So it's about 30 kilobytes, the genomic size or the genetic material size. Okay, finally remember 30. Coronavirus is grouped into all the following except the options are alpha cov. Cov means co for corona, V for viruses. Alpha covis, beta covis, gamma covis, or delta covis. Time starts now. In microbiology, coronavirus is grouped into three categories, that is alpha covis, beta covis, and gamma covis. So the except answer is there is nothing called delta covis or not there. Okay. The size of the coronavirus is, your options are 0.125 nanometer, 1.25 nanometer, 12.5 nanometer, 125 nanometer. Time starts now. The overall size of the virus is, most of you would have told this answer. I'll tell you why that is not the answer. It is 125 nanometer, because in most of the description they tell it is 0.125 micrometer. This and this almost appear similar, but answer is 125 nanometer. So it's better to remember this first option, the longest one. So it's a virus, right? So it can't be such a big size. So that's what you feel. Okay, initially you feel it cannot be such a big size. It can be either of these two. But here, these two can also be the answer. But you have to note this. After that number, what numericals will come? What values will come? 0.125 micrometer. Now nanometer and micrometer. In nanometer it's 10 to the power of minus 9. In micrometer it is 10 to the power of minus 6. So when you are reducing this 125 nanometer into micrometer, so it's nano, right? So you will make it into micro. So three digits you have to go like this, 1 2 3. So you have to put the decimal here now. See, that's why this is the actual size, 0.125 micrometer. I told you initially most of you would have gone for this because you'll think that this virus will not be that small, or it won't be this big, you feel, and you'll go for this option. But the correct answer is 0.125 micrometer, not nanometer. So this goes wrong. So it is obviously these two are wrong. So it is 125 nanometer. Kindly remember these two things, 125 nanometer or 0.125 micrometer. Come on to the next question. How many spikes does the coronavirus have on its surface? Is it 34 spikes, 54, 74, 94? Your time starts now. These are the spikes. Okay. How many of them are there? Can't count in 10 seconds. Just 74. You have to remember this number. 74. Just add 7 plus 4, it will become 11. If you write 11, it appears like spikes. So it has got 74 spikes on its surface. Kindly remember that number. It will be asked in the entrance exam. SARS-CoV spikes. Just now I was talking about the spikes. That spike glycoprotein has how many amino acids? Your option are 1155, 1255, 1355, 1455. So definitely it is 55. But which of these? Time starts now. It is above 1000 and below 1500. So kindly remember the option is option B, 1255. So just remember it's 12:55. That is in the noon time, 12:55, like a time you remember. That is the amino acids. Okay, that mid noon, because of maximum temperature, this virus spikes will get damaged. Just remember like this, 12:55. It is nowhere related to that, but for your easy way of memorizing, I'm telling you this. Okay, so 12:55 amino acid. SARS-CoV spikes glycoprotein has two domains, S1 and S2 domain. So it acts via which receptor? Options are AS1, AS2, AT1, AT2. So I'll help you out. SARS-CoV spikes are there. These spikes are glycoprotein. It has got two domains, S1 and S2 domain. Out of these two domains, the S2 domain acts via one of these receptor which is present in human cell. Is it angiotensin converting enzyme 1 or 2, or angiotensin 1 or angiotensin 2? The answer is AS2. So it has got two domains, S1 and S2. Through S2, it will act on S2. So S1 binds to the receptor, S2 binds to the transmembrane. So I'm sorry for that. S1 through the S1 receptor, it binds to the receptor. Through the S2, it's just a transmembrane portion. So kindly remember, SARS-CoV spike glycoprotein has got two components, S1 and S2. The S1 component binds to the receptor. The receptor name is AS2. Don't think S1 means AS1. S1 binds to the receptor and it binds to the AS2 receptor of the cell membrane. Spikes in the coronavirus is made up of, options are cold agglutinin, hemagglutinin esterase, warm agglutinin, hemagglutinin lyase. Time starts now. So the correct answer is hemagglutinin esterase. This is the component, this esterase component which causes hemagglutination, is present on the spikes of the coronavirus. Hemagglutinin esterase. Which protein of COVID means coronavirus determine the shape of the virus? Is it S protein, N protein, M protein, or E protein? Time starts now. The shape of the virus. If you thought it is S protein, it's wrong. S means spike protein. Okay, so it's determined by M protein. The M protein contributes to the membrane of this. So the shape of this, more often than not, it's spherical. It can also be oval like an egg, or it can be rod shape. So it depends on the M protein which gives the actual shape of the virus. Which protein of COVID helps in assembly, budding, and complete virus formation? Again, the options are same: S protein, N protein, M protein, E protein. Time starts now. The assembly means these proteins, all these proteins will come at one place, and the genetic material, that is mRNA, RNA, on replication it will form n number of RNAs, that will also come at one place, that is assembly. Then it will bud out of the cell, the host cell, and complete virus is formed. So which of this protein will help? The answer is N protein. So the N protein, N for nucleocapsid, the one which surrounds the nucleus, or the genetic material, genomic material, that is the RNA. This protein is important for these three steps: assembly, budding, and complete virus formation. Which protein of COVID helps in the attachment of the virus to the host cell surface? S protein, N protein, M protein, or E protein? Time starts now. You can tell this, it's easy. what is this? These are the spike proteins. How many are there? 71 are there. So these spike protein will have S1 domain. That S1 domain binds to which receptor? ACE2 receptor. So this is spike protein. S protein or spike protein is the one which binds to the host cell surface via ACE2 receptor. So S for spike. Okay. So spike protein. Which protein of course helps in the virus production and maturation? Your option are again same: S protein, N protein, M protein and E protein. The time is up. The answer is option D, E protein. So in summary, the virus has got 4 proteins: S protein, N protein, M protein and E protein. S protein is a spike protein. This spike protein, the S1 domain attaches to the ACE2 receptor on the host cell. So it is for attachment. The N protein is for assembly of the virions, the proteins of the virions and the genetic material, the budding and the new virus formation. M protein gives a shape to the overall virus, either it's spherical, oval or it is like a rod. So that is M protein, provides the shape to the virus. M means membrane, N means nucleocapsid. E protein, E for envelope. So this virus has got an envelope, the outermost covering. So that helps the virus for production of new virus and also maturation of that virus. So that is the purpose of E protein. So kindly remember SNME. S for attachment, N for nucleocapsid protein, M for membranous protein for the shape, E protein for virus production and maturation. It functions also at intracellular trafficking level, where it regulates the amount of genetic material produced and the proteins, various subunits which are produced. Viroporins are viral encoded membranes. So these viral encoded membrane are forming, definitely Viroporins form pore. But where do they form pore? Option: pore forming proteoglycan, pore forming proteins, pore forming lipids, pore forming phospholipids. Your time starts now. So what are Viroporins? The Viroporins form pore, but these Viroporins are what are they made up of? Is it protein? Is it lipid? Is it phospholipid? Is it proteoglycan? The answer is second option, pore forming proteins. See, this Viroproteins, virus is attack the human cell. It has to form pores on the surface of the human cell. So it should be a protein. It can't be lipid actually, because human cell is already made up of lipid. It can't be proteoglycan. So by default it should be proteins. Through the pores, the new virions will come outside the host cell and infect the newer host cells. Now to the next MCQ. Viroporins is anchored to the lipid bilayer. Just now I was talking. New host cell lipid bilayer by terminal positively charged residue of dash. Is it aspartate glutamate residue, tyrosine tryptophan residue, asparagine glutamine, lysine arginine? Time starts now. When the Viroporins comes outside now, it has got positively charged residue of which amino acid? The answer is the last one, lysine and arginine. So this lysine and arginine, these two amino acids has got positive charge on the surface, and this forms the terminal portion of the Viroporin, which is the receptor for coronavirus entry into the cell. Your options are metallolipase, metalloprotease, metallosaccharides, all of the time starts now. So the answer is metalloprotease. Just now I was telling, the virus is attached through the protein component. Viroporins have got protein. It has got lysine and arginine positively charged. So it enters also via protein component. It's called metalloprotease. Which is the co-receptor for the coronavirus entry into the host cell? The co-receptor other than the actual receptor ACE2 you already know. Other than ACE2, what is a co-receptor? Options: N-acetyl, cysteine, N-acetyl uramic acid, N-acetyl tyrosine, N-acetyl glutamate, N-acetyl glucosamine. So N-acetyl is common. These are the other options you have to look into. Time starts on. Time is almost up. Okay. The answer is N-acetyl uramic acid. How to remember? Short form NaNA. NaNA. The child is 6 months old, now it will call by bisyllable word like this. Na, meaning in South Indian language it means father, but just to remember, na. N-acetyl uramic acid is a co-receptor for the entry of coronavirus into the host cell. Coronavirus, after entering the cell, first produces, options are protein N, protein M, RNA polymerase, subgenomic RNAs. Now time starts now. If you want to understand this, kindly watch one of my video on pathogenesis of coronavirus. Okay, it is there in the description also. The answer is produces this RNA polymerase. This is the first protein the virus produces. As soon as the virus enters the host cell, it starts producing protein because directly it undergo translation, and the first protein to be produced is RNA polymerase. Why? It is an RNA virus, so it has to replicate itself. So it requires an enzyme. The enzyme is RNA polymerase. That's why. Okay. Later it produces all the other spike protein, nucleocapsid protein, N protein, membranous protein, E protein, and all it produces. Okay. Like and share this video with your family and friends. Don't forget to press the thumbs up button showing that you like this video. And if you're new to our channel, kindly consider subscribing to our channel. Don't forget to press the bell button to get the latest notification of the videos which we upload. Thank you once again watching Logic Medico and learning from here.